It's a great honour to speak to you this evening, and I'd like to thank the IES very much for that. My wife this morning asked me what I was doing, and I sort of said I was coming to give this lecture, and she said, where? And I said, oh, it's in the Royal College of Gynecology and Obstetrics. Then she got very, very stern and said, no medical puns whatsoever. So I'm not about to deliver a lecture to you. So what I'm going to discuss in this talk, and I'm going to not really do what Gary asked me to do and help you navigate. I'm really going to try and discuss some of these issues and sort of bring them up, so not try and give solutions. But to explore the different roles that scientists, sort of my community largely in research institutes and universities, how we contribute to policy making. And then I want to talk about a specific project that we're running out of the Oxford Martin School, part of the University of Oxford, which we call a restatement project. And I'll explain what that is, and I'll use that as an excuse to talk about two particularly contentious issues, the control of bovine TB and neonicotinoid insecticides, which I just call neonics. And then I'll finish off in the last couple of slides just by exploring some general issues about evidence going into policy and hopefully stimulating some discussion. And I put up those two rather cute slides, those cute pictures there, because I give a talk on bovine TB and policy. And at one point I had this slide up and I said, none of the issues in bovine TB are black and white. And one of the students put out their arms and said, I think you'll find it is, Professor Godfray. So what I'm going to do is I'm going to, first of all, talk about the different roles that scientists have. And I'm going to use a taxonomy that I rather like by a chap called Roger Pielka, who's written about this in, I think, a very interesting way. And he describes scientists as acting as four different roles: pure scientist, arbiter, advocate, and honest broker. And rather solipsistically, I'm going to go through each of them, the first three very briefly, and give an example of how I act in these different roles, and then spend much more time at the end on the honest broker. And it is slightly self-indulgent, but I'm a population biologist, I'm a professor of entomology, so I'm going to slip a little bit of the animals I love in, but just very briefly before going on to the main subject. So pure scientists. So Pielka says that actually many of us working in science, we actually think most of the time, especially when we're engaging with policymakers, that we're being the pure scientist. Nothing to do with policy, completely just, it's the facts man. And of course, it's very rare that we do that. Let me give you one example about, so I work in a department where we have people working on eagles and literally, and tigers and things like that. And I'm afraid I work on aphids. And I don't have the time to try and persuade you about the wonders of aphids. And we don't work on them because we're pests, we work on them because they have various things that you can do with them in the lab. And I'm going to give you one interesting fact about aphids, which is me being a pure scientist, which I don't think has any policy relevance whatsoever. Now, those of you who remember your A-level biology, all aphids have a bacteria inside them that synthesizes the amino acids that are not found in their diet. What we've discovered in the last 10 years is that any particular aphid clone, and they're asexual during the summer, has a smorgasbord of other bacteria, typically one or two. And these bacteria provide little goodies for the aphid. And we found, and this is my interesting pure science fact, we found that one helped you defend yourself against one of the main natural enemies of aphids, which is this fungal pathogen. So it's a horrible fungal pathogen, which in the presence of the symbiont still kills you about 40 or 50% of the time. But if you remove the symbiont by injecting with an antibiotic, then you don't get that, then you lose what protection you have. And this symbiont, we discovered, protects you against fungi. A group in the States found another protects you against parasitoid wasps, the other major natural enemy. Yet another protects you against heat shock. So we thought we knew quite a bit about aphids, you know, the major pest of temperate agriculture, but actually we didn't. So much of their phenotype, so much of what they do is determined by what they carry. Now, although aphids are press, I actually assert that I've talked a bit of pure science to you with no policy relevance whatsoever. So I hope you forgive me just showing you one thing I'm really enthused about, and my excuse is that that's me being a pure scientist. What about being an arbiter? This is, again, I think we in the science community rather overestimate our importance. You know, policymakers, they don't know whether to do A or B. They don't know whether to immunise now or immunise when you're 8 years old. You call upon the scientist to be the arbiter. Now, this does happen, and I gave the example of immunisation, that's a good example, but I think it's quite rare. In my career, it's only happened once. So I'm afraid this is another insect. This is an insect called the mango mealybug. It's quite similar to the mealybugs you probably have on your pot plants. But it was introduced from South America into Africa, where it attacks mangoes. An infestation like that has two effects. First of all, it means your mangoes are dried up and not very tasty. And second, those of you who travelled in West Africa in particular, every village in the middle has a big mango tree where all the social activity takes place under. And if your mango tree is covered with these things, dripping honeydew down, then it's not very pleasant. So you have an introduced insect. The classical thing to do is you go and you look for a biological control agent. And the International Institute of Biological Control, then the Commonwealth Institute, went into South America and came up with two insects. And these are actually the insects I love. So when I retire, I'll spend my declining years staring down a microscope at parasitic wasps. Now, I won't go into any of these details, but these are two small inserted wasps that had slightly different biology. They attack at different times of year. They have an intricate effect in intricate interaction with the mealybug, which is different. And IIBC needed to know which one to bring into culture. It would take about 6 months to bring it into culture. To rear it up, you have to go through all the regulatory hurdles. So they wanted to do one or the other. And they asked us to do a population model to say, was there any particular reason? And we did it. And again, I'm going to bore you with none of the details, but there were two parameters that we didn't know. Some things you can go out and measure. There were two parameters that we didn't know. One is the x-axis, one is the lines going from red, blue, green, brown. And this is population density, so you want to knock the population down. And you can see that there are some combinations of the unknown parameters that would make this one better, but for the vast, vast majority, this one was better. So we gave the advice, definitely go for the one on the right, called Anagyris. IBC said, thank God for that, we did it a month ago, we couldn't wait for you to do the modelling. So that is, I think, actually a genuine example of us being an arbiter. They're not, well, apart from that last slight wrinkle, that there wasn't a non-science component to it. Now, the other role that I think we have as members of the research community, both in and out of universities, is as an advocate. And we come with a particular set of skills. We as scientists, we often have hugely deep insight into a particular area. And so we can understand the evidence and incorporate that with the natural, economic, social science, the legitimate policy values. Now, when we do this, we're one of many people who are advocates who bring their own set of skills. So, for example, in many of the things that IES is interested, you'll be asking farmers, you'll be asking people who, fishermen, whatever. So we all bring different skills. And I think being an advocate is a really important role for a scientist, although we have to be clear when we're being an advocate. And I think one of Pielka's main points is that sometimes we confuse when we're being an advocate and when we're being an honest broker, and something that I'll come back to at the end. So I have one slide of an area that I'm an advocate and I feel very strongly about. So when I was asked to be chair of DEFRA Science Advisory Council, which is a great honour, then I was really keen to accept. And Ian Boyd, the DEFRA's Chief Scientific Advisor, is here this evening. He's just doing a fabulous job. at a very challenging time. And one of the things I said was, I believe that the chairman, that Science Advisory Council really needs to roll its sleeves up and to help DEFRA sort out its evidence base going into policy. But I said that there is one area where I will be an advocate. I will not be going into this. And this is Kew Gardens. I used to be a trustee of Kew Gardens. I feel passionately about Kew Gardens. It's one of the top 3 or 4 biodiversity institutes in the world. It gives us an enormous reach. I could go on. This is an example of where I'm being an advocate. And I have to be very careful with DEFRA because DEFRA is Kew's home ministry, that I don't confuse my role as a dispassionate sorter of evidence in SAC and my role as an advocate. And again, this I think was the important point that Pika makes, that being an honest broker is really hard, and it's something that you can probably never achieve absolutely, but it is something that is worthwhile striving to attain. And this is what we've been trying to do in the Oxford Martin restatement projects. And this is very much a co-project with my colleague at Oxford, Angela McLean, whose background is a mathematical epidemiologist. And what we want to try and do in the restatements project is to try and summarize independently the natural science evidence for a particularly complex topic. And our sort of target audience is someone not coming to the issue completely new, but someone who knows a bit about the area, but is not perhaps a technical expert. So a deputy director or director at a department, for example. And the aim is to be policy neutral. And again, this is an aim, but it's an explicit aim. And you call out and say, well, we're trying to be policy neutral. Tell us if we're not being. And in many ways, it's not aiming to do new analysis, but to take what is often a highly complex area and to distill it in a way that can be relatively quickly mastered by someone who, again, has some expertise, but is not a real strong technical expert. And so the output is a succinct summary plus annotated bibliography. I'll show an example of that in a moment. And what we also want to do is to state where uncertainty exists. And the way we've been doing this, and I won't read through these different ones, this is the particular one we use for the bovine tuberculosis case, which I'll talk about in a minute, is to have a sort of reserved vocabulary. And it sort of borrows some of the ideas that have come out of the medical world, things like grade and the Cochrane process, or out of IPCC, which has a particular set of terms. So again, we try and make it easy for people coming from the outside to assess the strength of the evidence, the consensus of the evidence base. So how do we go about it? Well, to a certain extent, we're making this up as we go along, but this is how we've done it so far. So in the projects we've done so far, we've identified a sort of super stakeholder, which it's tended to be the department that's responsible for this particular area. In the examples I'm going to show you tonight, it's been DEFRA, but we're doing projects for Public Health England and for the food standards agencies. I have to say this has sort of evolved. When we first did it, we just sort of started doing it without talking to the ministry first. And we heard back from the John Beddington, who was then the chief scientific advisor, who heard we were doing this, and I think was worried that this was going to be a work of advocate. And we heard back, I hope they bloody know what they're doing. Except he didn't actually use the word bloody, if I remember rightly. And we did have to convince the department at the time that we were honestly trying to be honest brokers. And I have to say that once DEFRA then really understood what we were doing, and delighted to see Helen Ghosh in the audience, who was permanent secretary there then. We had fabulous support from them in actually doing this. So we have a super stakeholder. We then recruit an expert group where we deliberately get people who have different views on a subject. We get them from the academic world, defined very broadly, not the stakeholder NGO industry world, because we want to have a conversation in detail about the evidence. And so we use that to draft a restatement. We have a meeting. We're so lucky in Oxford to be able to hold meetings in places like that. That's All Souls. And then we come up with a draft of the restatement. And then we then do what we call stakeholder consultation. So we send it out to typically 50 or 60 people from the industry, from the NGOs, from academics we didn't invite. So those of you who are regularly write academic papers, and you know when you have 2 or 3 referees' reports land on your desk and your heart sinks, when you get 60 referees' reports from this, but they are really, really helpful. And especially as we're asking the stakeholders to say, are we being, is this correct? Do you agree with it? Have we interpreted the evidence? But also, are we being policy neutral? I mean, it's very interesting in the insecticide one, which I'll show you in a moment, that both the agrochemical industry and some of the environmental NGOs came back on different sentences and says, well, hidden in that sentence is an implicit assumption that insecticides are good or insecticides are bad. And that was so helpful just to get our language right. And again, we'll never achieve perfect policy neutrality, but we think that by putting that out as a goal and asking people to call us out if we fail, that's one way of doing it. And then we then go through iterative revision with the expert group, and then we get it externally peer reviewed. And the ones we've done so far have come out in the proceedings of the Royal Society. And so it comes out, again, so far in this journal, in a rather curious way, and full credit to the Royal Society for being flexible. So it comes out a really short paper, typically half a page or a page, which you have to have a paper because it's a paper. And then the restatement comes out as the appendix of the paper, and it's a series of numbered paragraphs like that, which again, we're sort of having our mind's eye, the Director General, the Director, the Deputy Director, the head of a charity or something like that, who really just needs to try and get to grips with that subject. And then there's an annotated bibliography. So all the stuff you cannot shoehorn into a succinct paragraph with all the references goes in there, and then you have the references at the end. So you get a sort of package which is made up of 4 components, all based around the restatement, which again is the thing that we hope is going to be most useful. So let me just briefly describe the 3 that we've done so far, 1 on bovine tuberculosis and 2 on neonics, curiously, and I'll explain why. And in the sort of 10 minutes I'm going to give to each, I really can't give justice to it, either the science or the complexity of the issue. And I know that some of you will be, I'm looking at Helen there, will be extraordinarily familiar with these issues. So this is bovine TB, and actually I think this is the graph that sort of shows what we're having to deal with. The hot colours are the spread of bovine TB through the UK. That goes up to 2010. It's declined very slightly. Well, the rate of increase has declined slightly, but it's still a very major problem. I'm not going to go through everyone here just to save time, but we had the A team. You could not want a better group of academics who know this area backwards and forwards, who have very different views. And I have to say that the day when we get lock these people in a room in All Souls, give them a lot of food and drink and things the night before and things, it's just intellectually fun. And it's also interesting that in all the ones we've done so far, where we have people from different fields, the first sort of 20 minutes is sort of sizing each other up and sort of saying, well, how are we going to do this within the advocacy world? And then Angela and just bang on and saying, no, we're not trying to do advocacy, we're just trying to sort out the evidence. And then so far, one day it'll go horribly wrong, I'm sure, so far everyone's got it. And then they have been some of the most intellectually exciting days of my life. So we're not doing this out of any public spirit, it's just fun for us to do. So why is bovine TB difficult? Well, the tests for the disease it's just a difficult disease to detect. It hides away in cells. The tests have limited sensitivity. If you can imagine a more hard wildlife disease, you would have a social animal that lived underground and came out at night. I mean, it's really hard, and there is little doubt that badger is, well, there is no doubt, I think, that badger is the major wildlife reservoir. And the transmission and biology of the disease is very hard to understand, is complicated, and we don't know enough about it as much as we'd like, despite fabulous research. It's just so hard disease. And of course, there are all the political complexities of this disease involving one of the iconic species of the British countryside. There are many areas of contention. Why is it spreading? Why are some farms hotspots? Why, if you clear a herd, does it come back sometimes later? Actually, we're beginning to get a better idea of that. How do you get the test better? We believe that biosecurity works, just putting cattle troughs high enough that badgers can't feed from them. We don't actually have any evidence for it, just because it's really hard evidence to get. Cattle movement restrictions, that affects farmers' livelihoods and income. Lessons from other countries. How much can we learn from, example, possums in New Zealand? Forgive me an anecdote, I'm half New Zealand. I have no farming relatives in the UK. All my New Zealand relatives farm, and they're dairy farmers, and they worry about possums. But possums is this horrible Australian animal that was introduced to New Zealand. I have a first cousin, a lovely man, Stephen, laconic. He was driving me between two aunts, and we sort of lapsed into silence as we drove through the New Zealand agricultural landscape. And I think I was dozing off, and he suddenly swings to the right, and there's this bang. And this large furry thing hits, we're in a farm vehicle, I should say, hits the windscreen, blood everywhere. And I go, Stephen, I thought we'd hit someone, and things like that. And he goes, flipping possum. Interestingly, he used the same word as John Beddington, not to... We don't know enough about vaccination and how it works, either in cattle or badgers, and of course the big issue of shall we cull badgers. And of course, we've been worrying about this for a very long time. And 15 years ago now, John Krebs, my current just retired boss at Oxford, did the Krebs report and recommended, and full marks to DEFRA for carrying it out, one of the biggest ever experiments that has been done, the so-called randomised badger control trial, where triplets of sites, 10 triplets of sites throughout the west of England were put into 3 treatments. A control where you went out and culled, and where you only culled when there was a case of bovine TB, and which was abandoned for reasons which I'm happy to talk about if people are interested. This was a massive experiment. It lasted 10 years. It cost a lot of money. It was beset by problems, so people trying to sabotage it. Foot and mouth came out in the middle of it. And I won't go through this graph in any detail, but that is what happened to... So you're culling badgers. This is a period of culling. And if you just look at the black line, it's going down, and it stays down for a bit and then comes up. So it does seem culling does have an effect, but the red line is the peripheral area. And so in peripheral areas, you did get some extra cases of TB. But this is evidence of culling. I mean, my view of this is that if you cull badgers, you will get an effect, but it won't be a large effect. And so what we try and do in the restatements, we don't come out, and it's frustrating. Journalists say, well, you've done this, what is the result? But that's not the point. We're trying to say, what is the evidence landscape? What is the evidence landscape upon which people can make decisions or recommendations that may be different depending on the values they accord to different stakeholders, the farming community versus the environment? And I'll come back to that at the end. So it doesn't provide answers, but what we hope is it provides a level playing field that we can talk about what really matters, which is often the values and the politics. So let me go through the second example here, which is neonicotinoid insecticides. So neonics are a major class of insecticides. They are relatively new. They came onto the market in the 1990s, and they now, this is slightly out of date actually, they're now over 30% of the insecticide market by value. Now, wild pollinators have been declining, and honeybees have been declining as well for very many complex reasons. One of the most important has been this evil-looking mite that has spread throughout the world in the last 20 years. So we've had a decline in pollinators. We know other things that have been happening, especially with honeybees, but we've had this introduction of a new insecticide. And why this insecticide is different is that it is used as a seed treatment. Very, very small quantities of the insecticide get into the plant, spread systemically, and protect the whole plant from herbivores, insect herbivores. But they also get into the pollen and nectar, and so they can conceivably, or so they can, affect pollinators. And there was sufficient concern about this that in 2013 the EU brought in partial restrictions on neonics, so that they cannot be used on plants that produce flowers that are visited by bees. So you can't use it on oilseed rape or sunflower, you can use it on wheat. So here's our group. Again, people coming from very different views, but again, in my slightly biased view, this is an A team that you would want on the panel. So forgive me, here's my nerdy slide of the talk. This is sort of honeybee economics. So this is a type of argument you can make. So actually that's a bad thing. That's a willow cack in there, but suppose that's an oilseed rape for the sake of argument. The pollen and nectar in oilseed rape would have typically 2 to 6 parts per billion, nanograms per gram in there. So that we can go out and measure, and that's typical. There is some variation. And then we also know from lots and lots of lab studies that honeybees, if you give them about 3 or 5 nanograms, then they keel over. Then you can sort of work through the sort of math. So a foraging honeybee, so that it collects 32 to 20 milligrams. That will contain that amount of insecticide. So how much do they eat? Well, it depends what type of bee. A nursing honeybee, very appropriate for where we are this evening, can consume 7.2 milligrams per day of pollen. So they largely eat pollen when they're nursing. So that's only 1% of LD50. A nectar foraging honeybee can get about 7%. And this sort of is probably a worse case because they would be feeding on a more diverse group than just oilseed rape and just field data. So in many ways, and this is why the insecticides were allowed in the beginning, this is quite reassuring. You would only expect sort of 1% of LD50, 7% of LD50. You would not expect to get much effect. But actually, it's more difficult than that. And what led to the introduction of the restrictions was that evidence began to mount that there were chronic effects, so building up, that there were sublethal effects as well, so that even relatively small amounts of insecticide, of the order of 5% of LD50, could interfere. Well, they're neurochemicals, so you expect them to interfere with bee behavior. There was also data that there was, there were interactions with other stressors, in particular Varroa, in particular viruses, and with other insecticides. Not great data, but there was some data out there. We were worried, or the community were worried, that actually most of our data came from honeybees. We knew far less about bumblebees and virtually nothing about the other bees as well. And we were very unclear about whether our lab data extended to the field. And I won't go into the complexities, but we had semi-field experiments, but we had no proper field experiments where you did replicated studies of putting bees in different landscapes and seeing how they were affected. So there was a huge amount of unknown. And many areas of contention, so the role of insecticides. It's clear that that bee populations were going down, species richness was going down, but probably been going down for 100 years. Insecticides, neonics were only about quite recently. There's evidence about abatement recently, that the rate of decline has fallen off. And honeybees, well, honeybees, I mean, what largely determines the number of honeybees is the number of beekeepers and their motivation. We knew very little about actually so even if honeybees were going down, was this reflected by less pollination? We had very hard to interpret what the sublethal effects meant. Were they compensated for? And the experiments that then were then available were difficult, had many difficult problems. So exactly how realistic were the doses used? Exactly how were they analysed statistically? So what we did in the restatement that we published 2 years ago was to explore some of these issues. And to be honest, we then thought that we would move on and do something else. But this year, partly because the restrictions are coming up to review, partly because new data became available, in particular a really neat study by a group in Sweden led by Madge Rundloff, the first big, properly replicated real-field experiment. Mark Walpole asked to do a slightly smaller study to bring the restatement up to date. And essentially, this is what we concluded from that. We said there was pretty much a consensus that bees will frequently encounter neonicotinoids in fields at doses unlikely to kill sometimes, but largely have sublethal effects. And the big issue is how does that transfer first into population effects and beyond that into ecosystem services? And this is the new data that was beginning to come through. And we concluded that actually it was still too few data to be in any way definitive, but the emerging consensus was that honeybees were less likely to be infected than bumblebees. And there's a couple of papers which will be published later in this week that are still embargoed, that taking this story a little bit further. And the final thing we pointed out, and this is actually one of the sad casualties of the age of austerity, is just collecting the data on what farmers do in response to these bans. We don't know if farmers, if they haven't got neonicotinoids, then go and do something that is more environmentally benign, more pollinatively friendly, or whether they perhaps go back to use worse insecticides that they'd used previously. And I think we'll get some data on that soon. So this one we launched for the first time at the Science Media Centre, the unit run by Fiona Fox in the Wellcome building, which I think does a great job. And we actually had a really interesting interaction with the journalists who first said, well, what would you recommend? And so we said, no, we're not recommending things. And then it's great, they got it. And we had 2 really good articles that were published that we were really pleased with. One was by Damien Carrington in the Guardian. And look at the headline there, neonicotinoids, new warning on pesticide harm to bees. David Jones in Farmers Weekly, no sign of damage. Now, well, your first reaction, it's my first reaction, is to laugh. But actually, it was great. They both got the science absolutely right. And Carrington in particular explicitly then said, well, based on this science, we introduce our value judgments, our belief about the precautionary principle by things, and then came up with the different conclusions. And both Angela and I were absolutely delighted by this. I hope we weren't deluding ourselves, as an example of where we believe being an honest broker is doing the right thing, putting a level playing field. So then you argue about what really matters, which is the value judgments and the things that politicians really need to, where politicians have to come in to make decisions. So just briefly, how am I doing for time? Just briefly, these are the restatements that we have on train at the moment. The next one that will come up will be on the low dose effects of radiation. And I can't resist, so Angela is leading on this one, but this is an extraordinary bit of science for me. I hope you find it interesting. So this is cumulative mortality from lung cancer as a function of the amount of radon in your home. So if you live in the West Country, there's a lot of radon in your home. This is a lifelong non-smoker, and it makes really no difference having radon in your home. If you're a smoker, then you really suffer from radon, so there's an effect. And isn't this a fascinating thing? If you were a sort of Bentham utilitarian, you would then go into each house and say, do you smoke? Yes, you do. Right, I'll radon-proof your house. Do you not smoke? Then I won't. Now imagine trying to get that over as a policy. So it's been fascinating over the last 2 years reading 300 papers on the effects of neonicotinoids on bees and things like that. Now, what I'm about to say is going to sound pious, and I really don't mean it to be, because I, like all of us, when I do things, I suffer from unconscious bias, confirmation bias. But when you read those papers, the confirmation bias that you get, the unconscious prejudice, is just enormous. And you can see it, and it's not evil, it's just the way we are built. You know, if you're campaigning against something, you look at data in one way. If you're campaigning against the other way. And I don't know how we cope with that in the scientific literature. It's unavoidable. My view is we have to be far more clear about what that is and acknowledge it, and acknowledge that occurs as much in studies that we do in academia funded by a research council as in an industry study. Much of the confirmation bias in the subconscious bit, I think, is completely biased. I think there is what Pielka also calls stealth advocacy in that, which is actually, in the guise of being an honest broker, pushing a particular line, and it could be one or the other. And again, I think we just have to be aware of it. Something that was quite interesting is, in the first restatement, we said neonicotinoids are an effective insecticide in oilseed rape and a few other things, and put down a whole load of references. And a very smart chap in an NGO said, well, you've done this, but all the evidence is industry funded. And we looked, and we couldn't find any evidence that wasn't industry funded, for very good reasons. Why would a research council or equivalent body fund research that is only going to be used to a commercial company? So we have a real issue at the moment, is that how do we allow industry to do experiments that can then be more broadly trusted? And I believe strongly that we have to look at lessons from pharmaceuticals, which has come a longer way, perhaps from a slightly worse start. And they have things such as trial registries. So you say you're going to do an experiment, you set it up, you sort of announce you're going to do it, and so you can't hide the results, or you can't not publish a result that doesn't go your way. I think industry, which is on the back foot in many of these things, could gain tremendously by going onto the front foot and voluntarily doing some of these things. Sorry. I'm not certain that the way we regulate agrochemicals is sensible at the moment. I think that the precautionary principle, which if you interpret it, in my view, in a sensible way, that you have to be very cognizant of what statisticians would call tail risk, is really, really sensible. But if you interpret it as just anything that we don't know 100% about, you can't do, that makes no sense. We have to do it in terms of regulatory comparative risks and things. I suspect in this audience, I don't have to say that the linear model of evidence-based policymaking, none of us believe that anymore. The old idea that natural and social scientists did their work, came up with evidence, you then gave it to a civil servant who gave it to a politician who then enacted it. That's hugely more simplistic. I think it is really important, and sometimes we in the science community, when we're in advocacy mode, we do not realize that what's important is, I don't think it's as much evidence-based policy making as evidence-consistent policy making. You can have different policies that are consistent with the evidence. Hilary Benn, when he was in charge of badges, he said something which has really struck me. He said, you can have all the evidence that you can give me as much evidence as you like that capital punishment reduces the murder rate. I still will not bring it in because I believe state killing is wrong. And to me, that's perfect, in that even though the evidence would say one thing, you're clearly saying why you're disagreeing with it. And I think that you can have multiple in both the examples I've given this evening, you could have different things that are consistent with the evidence. Can I go on for, that was 40 minutes, can I do another 2 minutes? And that's really the critical thing. And there are some real things that sort of push in other ways. So bovine TB, neonics, these are just lose-lose for any politician. You know, there is nothing to be gained for being radical in those two things. It is far too simple for them to hide behind science, and we do everyone a disservice as scientists if we say you can hide behind science and that there is a linear model. And I go back to the point that I think Pielka made, and I strongly agree, that there are multiple roles for scientists. And I'm really not stressing that being an honest broker is better than being an advocate. When it comes to Q, when it comes to climate change, I really am an advocate. But I think you just have to be clear about those two roles. And I think my community, all of our community, is that we should be aware of misusing the authority of science. I mean, science is different. I mean, there are many different belief systems. When it comes to, I love my wife, I don't want a scientific explanation for that, but I do want a scientific explanation which will tell me my plane will fly to wherever. Science is different. It has a unique authority over any other way of knowing. And as scientists, we mustn't misuse that when we are integrating the evidence with our personal value judgments. But that's not an excuse to say, that's not saying we shouldn't be doing that, but we should be clear when we should be doing it. And very briefly, I think one of the other issues, and here I'm also quoting Pielka here, who says, well, is there a role to go beyond the honest broker of the science evidence to the honest broker of policy options? Now, I used to be a little cynical about open policy making, but I was lucky enough to be on the steering group of the Green Food Project. That was Jim Pacer's baby when he was Minister of Farming. And you get people in a room, you get a minister there to sort of make certain everyone behaves, and you can get really fascinating discussions between, say, industry groups and NGOs and things. So I think there might be a role to go beyond the purely evidence-based to trying to set out the alternative policy options. Because in some ways, both bovine TB and neonicotinoids are almost intractable within the boundaries that we put them. Perhaps with BTB, bovine TB, we actually have to ask difficult questions about the structure of farming in the Western Wales. I mean, largely we're doing it because we've always done it like that. For example, in neonics, then maybe we don't want to ban them everywhere. We may want to ban them in some areas, but not others. Sorry, that's a bit faint there, but that's the making the, I've just forgotten the name of them, the nature improvement areas that came out of the Lawton report. And I know John is your president. So, sorry, I've gone over 5 minutes. Thanks very much, and I hope that sort of stimulates some discussion.
Helen Ghosh, now at the National Trust, formerly at DEFRA. It was just wonderful to be taken back. I'm glad I didn't have to do neonics, but I've done radon, I've done bovine TB. In a sense, you kind of stole the question I was going to ask. I might have quoted, I remember the quote from Hilary Benn, because I was there then, and I thought that was a very powerful explanation of the role of the elected person in taking decisions. I'm really interested in your point right at the end about the scientist and their role in posing the bigger question. I remember, and I think this doesn't go against my confidentiality sign-up, talking to the now Cabinet Secretary, Jeremy Haywood, when Hillary and the department were faced with, as it were, that round of decisions about badger culling, when he said, but sorry, what is the problem? And what is the problem we're trying to solve? And you know, of course, in some ways, given pasteurisation, there isn't actually a public health risk. It's very much determined by the rules we happen to have in Europe about vaccination when we get a proper vaccine. I won't ask you when we're going to get a proper vaccine. It's a kind of socially constructed problem, which is not really, if you could move to vaccination and animals going into the food chain, where is the problem? So could you just sort of say a little bit more about whether, I don't know, whether you and your colleagues in Oxford or elsewhere have a kind of ambition to say you are, in an honest broker sense, people who could ask the big question in a way that perhaps policymakers and even NGOs such as the one I now lead can't ask it. Could you just say a bit more about that? Because it's actually going back to the real problem.
Yes.
Is what lies behind a lot of these policy angsts, if there is such a plural.
Gary, would you mind if I answered that one sort of straight up? I will say slightly facetiously, you know the answer to when will vaccines become available.
It's always 10 years away, I know. We're helping with the can you administer the vaccines on some of our land, but it's always 10 years away.
I think you make a really interesting and important point. I think, so I'm not talking now about the whole research community, but my area of the research community is the HEIs and things. I think that I don't think we help you at the moment. We are stuck in our silos. So I'm in a zoology department, other people are in the economics department. I think the good news is that in many of our major universities, we're wondering how we can work together in different ways to actually try and make it easier to answer that type of question. So I lead something in Oxford called the Oxford Martin Programme, The Future of Food. And it's, in fact, Chris Gilligan, who's at the back there, does something similar in Cambridge. And it's not just Oxford, Cambridge, many other universities, I think, are trying to get into a place where we can do things such as that. And I think working with RSBB, National Trust, CLA, NFU, we should actually, unless we engage with organisations like that, then there's a risk that we'll go off into tangents and blind alleys in our ivory towers.
Okay, thank you. Excellent. So there's this gentleman here with the beard.
I have a comment and a question. My question is about the precautionary principle. You alluded to how it can be misused. What do you think we can do to prevent this from happening, to prevent it from being hijacked as a campaigning tool to prevent innovation, perhaps? And my comment is about, you held up the pharmaceutical industry as good practice.
Not for everything.
Well, I would just say on clinical trial reporting and registration, the best available evidence suggests that only about half of clinical trials have actually been published. Many haven't been registered. If that shocks you and you think that's not the right situation, then I suggest you go and sign the All Trials petition to change that.
Sorry, could you just get your name?
Chris Peters from Sense About Science.
Okay.
Well, that's a great point. And actually, I know that you and Sense About Science have been arguing for this, so more part of your elbow. I think you'll know more about this than me. I think things are getting better, but you're exactly right. Sorry, what was just remind me your first point again? Yes. Well, sorry, I'm going to be a bit nerdy as a mathematical biologist, and I think one needs to start with what one can say quantitatively about the alternative options, but then not believe. It's more complicated than just putting a probability distribution around it. There's a lot of modern ways of doing that, and I think there's some really good practice. Where precautionary principle is really good is it makes you think about the tail risk and things. I refuse to use the word black swan.
Thank you. Right, there was a chap up here. Thank you.
Hi, I'm Michael Warhurst, and I head up an organisation called ChemTrust, a charity that works at a lot at EU level on chemicals policy issues. I think my question is really about the scale of the job, because with neonics you're dealing with a few chemicals and a few insects. The endocrine disruption report you're talking about, there's an awful lot of chemicals in debate, and there's an awful lot of different endpoints and an awful lot of different organisms, and there's many 150-page plus reports on it. How do you think you're going to be able to address something of that scale?
We're grappling with these things right at the moment, and exactly as you say, it's really complex. Endocrine disruptors are defined by an effect rather than as a chemical class. I can't tell you whether we're going to be successful. Come back, well, if I could have your card later, I'll recruit you as one of the stakeholders to give us a hard time.
Very good. Dougal.
Dougal.
Hello, I'm Dougal Goodman from the Foundation for Science and Technology. Thank you for a brilliant talk, which I have heard part of it before.
Sorry, Dougal.
But what it struck me is the issue of trust. And I remember when I heard it before from Angela McLean rather than yourself. She talked about how you'd done the restatement paper, but then a new paper emerged from Sweden, which changed the opinion. The decision maker is making a binary choice. Do I, don't I do it? And is looking for advice from the science community. But science is moving along all the time. So there can be a loss of trust when the decision maker says, well, I thought last week you told me this. But now this week you're telling me something else.
Well, remember what we're doing is we're summarising the evidence at any one time. So it's not that... So the Swedish paper you mentioned is the Rundloff paper that actually spurred Mark Wolpert to ask us to do the second neonicotinoid study. It is so the evidence base evolves. It's not that, or very rarely, that previous evidence is wrong. It sort of goes. And remember, we're not advocating policy.
Sure, that's the point I'm trying to get at, is the minister is making a binary choice, makes the choice based on often incomplete evidence.
Yes, so we're being very clear what we see our niche in that hugely difficult decision, which is to clarify the evidence base such that he or she can then better assess the arguments of different stakeholders. For example, in the neonics case, maybe the industries and some of the NGOs, and that his or her civil servants advising him or her it makes it easier to look at the evidence base. And I think our contention, which is, as Angela at your wonderful meeting said, that so often the science is used as a proxy argument for what are often valued decisions. And what we are trying to do is to have the discussion and the legitimate arguments in a democracy where many of us have different worldviews, is to try and have that argument not about the science bits, which should just be, you could be arguing A or B, they need to be consistent with the science, but actually to have the argument about what it's really about, which is very frequently values and worldviews.
Very good.
Is that answering your question?
Lovely.
It's developing the argument, yeah.
Right.
Very good, thank you. Chris Gilligan.
Chris Gilligan from Cambridge and chair of JNCC. Great talk, fascinating, and lots of questions come to mind. But one in particular for me, you talk about the evidence base, and underneath that, Charles, of course, is the scientific method and inference, hypothesis testing, statistical methodology. The question then comes, do we understand how we should be using our statistical methodology? And do we have the right methodology available for what we need to do?
I think we probably have better methodology than we typically use. This goes back to Dougal's question a bit, that one cannot say, I'm not going to summarize the evidence now, I'm going to wait for 15 randomized controlled trials to come. I think that there is a duty on our community when we're acting in honest broker mode to actually see what's out there, and that will be using the full panoply of modern statistics, but it will be also using some of the more less curious, less standard ways that, for example, at your university, David Spiegelhalter has championed, and some of the techniques that come out of the social sciences as well. So we don't have the luxury of just saying, go away and do an experiment until we can do the proper statistics. I think part of what we need to do is to summarize the evidence as it exists, but then be rigorously honest, which is the reserved vocabulary we have and the different ways we classify the results. Just as a sort of slight aside, we started doing this because Angela and I were having a discussion about bovine TB, and with her medical background, she said, well, why don't you do a Cochrane or a Grade type study? So we sat down and started going through the evidence, and it turned out that everything, all the bits of evidence relevant would have come in the very lowest grade, or lowest category of grade. Now, that's nothing to be ashamed of. I mean, Medics, speak it quietly in this building, have it easy because they have plenty of replicates. I mean, you saw the randomised control trial there. That was a big experiment with 10 replicas.
Thank you. I enjoyed that talk very much. My name's Carolyn Roberts. I work for the UK Knowledge Transfer Network. I've just come here, actually rushed here, from sitting in a meeting in Westminster that was discussing the Birds and Habitats Directive and in the context of sort of red tape challenge, that kind of thing. And actually there was no mention of science at all in the discussion. I found the discussion actually almost impenetrable, I have to say. But I was just wondering how the characteristics of the debate that you've introduced us to, which I found very compelling, actually play out in a multinational context of something like the EU. Because there it seems to me we have, well, some of the, what you called those roles for scientists seem to me to be automatically playing out in differently weighted ways in comparison with how they would play out at a national level.
Yes, you're interesting. That's a very interesting point, that there's subtle, almost cultural ways in which science advice works in different countries. I'm not saying the Pielka taxonomy is the only or even the best one. To me, I find it helpful in just thinking about the different roles, and you can perhaps come up with something more sophisticated or better. But to me, I think we as scientists giving advice and things need to think about whether we are trying to be dispassionate and whether we are trying to be advocates. And when we're trying to be dispassionate, to realise you can never be completely dispassionate, but you're putting it up and sort of challenging people. But yes, I don't have a straight answer to your question beyond acknowledging I agree with you, the differences.
Hi, Robert Ashcroft from the IS. You talked about needing to be aware of confirmation bias and stealth advocacy. Do you think that in our institutions we actually encourage young scientists early in their careers to think about the roles they're playing when they're talking about their own work?
No, and we are talking about this in Oxford to actually try and do that. One area that we now are good at, well, saying good is probably wrong because we're moving in the right direction, is unconscious biases during interviewing, especially when it comes to gender, race and things like that. So we make our people go on courses which get you to just think. Now, that doesn't guarantee good behaviour, but I think it's helpful in that. I think it would be useful for just when we write up our papers and things. I do wonder whether we have our incentive structure right in science. I sometimes wonder that some of the journals don't have the right incentive structure as well, and that they can, in a way of, in an age of impact factor competition, then they can often go try and push for a more sensational story than actually exists. I would like to see in the areas I've talked about this evening and in related areas, journals being more willing to perhaps say, well, submit your experimental plan, have that refereed, and we'll guarantee publication as long as you've carried out the experiment to a certain standard, irrespective of the result. I'm trying to persuade the British Ecological Society journals to do that, and that huge success so far.
Excellent.